Question number 151. In the cloning vector PBR322 given above, the restriction site marked X is possibly. Now, tetracycline resistance gene contains sites for BAMH1 and SAL1, whereas ampicillin resistance gene contains sites for PST1 and PVU1, and here is a rope gene that has sites for PVU2. And the X may represent either BAMH1 or SAL1. So the correct answer to this question is option number 1, SAL1. Option number 1 and 2 are incorrect because these are the sites present in ampicillin resistance gene, and option number 3, PVU2, is incorrect. It is present in rope gene. So the correct answer to this question is option number 4. Let's move on to the next question. Question number 152. While isolating genomic DNA, purified DNA can be ultimately precipitated out by the addition of, and this can be precipitated by the addition of chilled ethanol. So the correct answer to this question is option number 3, chilled ethanol. Option number 1, proteases, is incorrect. Proteases are enzymes that will digest proteins. Option number 4, ribonucleases, is incorrect. Ribonucleic acids. And the last option, calcium chloride, is incorrect. Calcium chloride increases the efficiency with which DNA enters the bacterium through pores in its cell wall during transformation. So the correct answer to this question is option number 3. Let's proceed to the next question. Question number 153. Which of the following is not a component of reaction mixture of polymerase chain reaction? And the very first option given is 2 sets of small chemically synthesized oligonucleotides not complementary to regions of DNA called probes, and this is an incorrect statement, as well as it is not a component which is required in a mixture of PCR. So this is the correct answer to our question, because in PCR mixture 2 sets of primers and not probes are required, and these are the 2 sets of small chemically synthesized oligonucleotides which are complementary to regions of DNA. The next option is a thermostable DNA polymerase, as it is a component of reaction mixture of PCR, so this cannot be a correct answer. Third option is deoxynucleotides to extend the new DNA strands. This is also a component, so cannot be a correct answer. The last option, small amount of DNA to serve as initial template, is also required, so this cannot be a correct answer. And the correct answer to this question is option number 1. Let's proceed to the next question. Question number 154. Select the incorrect statement with respect to EcoR1. And the very first statement given is, it is an endonuclease that breaks sugar phosphate backbone of double-stranded DNA. As it is a correct statement for EcoR1, this cannot be a correct answer. The next statement says it is an endonuclease that breaks phosphodiester bonds of a DNA molecule. As it is also a correct statement, this cannot be a correct answer to our question. The next statement says it is an exonuclease. It is an exonuclease that cleaves monophosphoester bonds. This is an incorrect statement because EcoR1 is an endonuclease. So this is a correct answer to our question. The last statement says it is a nuclease that recognizes specific palindromic sequence, is also a correct statement with respect to EcoR1, so cannot be a correct answer. And the correct answer to this question is option number 3. Let's proceed to the next question. Question number 155. Alkaline phosphatase is employed in genetic engineering as it helps to. The very first option given is remove terminal 3' OH groups of a linear vector, and this is an incorrect statement because alkaline phosphate is employed in genetic engineering as it helps to remove single phosphate groups from 5' end of linear DNA molecules. So second option is the correct answer to our question with respect to alkaline phosphatase. And why do so? It do so to prevent recircularization. This is the correct answer to our question. Alkaline phosphatase removes single phosphate group from 5' end of linear vector to prevent recircularization during cloning or to dephosphorylate DNA. The next option is add nucleotides at the 3' end group of circularized DNA is also incorrect. The last option, add single phosphate group at 5' end of linearized DNA, is also incorrect. So the correct answer is option number 2. Let's proceed to the next question.